Locomotion and movement. NCERT based single sentence questions. Let's start. One of the significant features of living beings is movement. Dash and dash exhibit a wide range of movements, animals and plants. Dash in the unicellular organisms like amoeba is a simple form of movement. Streaming protoplasm in the unicellular organisms like amoeba is a simple form of movement. Protozoan group with locomotory organelles as cilia are ciliates. Protozoan group with locomotory organelle as flagella are mastigophores. Phyla with tentacles, Cnidaria and Ctenophora. Human beings can move with the help of limbs, jaws, eyelids, and that human being can move limbs, jaws, eyelids, and tongue. Some of the movements result in a change of place or location. Such voluntary movements are called locomotion. Walking, running, climbing, flying, swimming are all some forms of locomotory movements. Locomotory structures need to be different from those affecting other types of movements. For example, in paramecium, dash helps in the movement of foot through and in locomotion as well. In case of paramecium, cilia are helpful. Paramecium cilia helps in the movement of foot through cytopharynx. Next, locomotory structures need not be different from those affecting other types of movement. For example, hydra can use its tentacles for capturing its prey and also use them for locomotion. Human use dash for changes in body postures and locomotion as well. Human use limbs for changes and body posture and locomotion as well. Dash and dash cannot be studied separately. Locomotion and movements cannot be studied separately. The two may be linked by stating that all locomotions are movements, but all movements are not. All locomotions are movements, but all movements are not locomotions. Methods of locomotion performed by animals vary with their habitats, and the demand of the situation. Dash is generally for search of food, shelter, mate, suitable breeding ground, favorable climatic condition, or to escape from enemies or predators. Locomotion is generally for search of food, shelter, mate, suitable breeding ground, favorable climatic condition, or to escape from enemies or predators. Cells of the human body exhibit three main types of movements, namely amoeboid, ciliary, and muscular. Amoeboid movement, ciliary movement, and muscular movement. Specialized cells in our body which show amoeboid movement, macrophages and leukocytes. Cytoskeletal elements which are involved in amoeboid movements. Are microfilaments. Cells of human which show locomotion by pseudopodia formed by the streaming protoplasm as in amoeba, macrophages and leukocytes. Ciliary movement occur in most of our internal tubular organs which are lined by the ciliary movements occur in most of our internal tubular organs which are lined by ciliary epithelium. The coordinated movements of cilia in the trachea help us in removing particles and some of the foreign substances inhaled along with atmospheric air. The passage of ova through the female reproductive tract is facilitated by this kind of movement, ciliary movement. Movement of our limbs, jaws, tongue, etc. require dash movement, muscular movement. Dash property of muscles are effectively used for locomotion and other movements by human being and majority of multicellular organisms. The contractile property of muscles. Locomotion requires a perfect coordinated activity of muscular, skeletal, and neural. Muscular, skeletal, and neural. Cilia and flagella are the outgrowths of the cell membrane. Dash movement help in swimming of spermatozoa. Spermatozoa, flagellar movements. Dash movement helps in the maintenance of water current in the canal system of sponges. Flagellar movement. Dash movement helps in locomotion of protozoans like euglena. Flagellar movement. Movement with flagella. Muscle is a specialized tissue of mesodermal in origin. Mesodermal in origin. About dash percent of the body weight of human adult contributed by muscles, 40 to 50%. 40 to 50% of the muscle. The four special properties of muscle are excitability, contractibility, extensibility, elasticity are the four properties of muscle. Muscles have been classified using different criteria, namely location, appearance, and regulation. Based on their location, three types of muscles are identified: skeletal, visceral, cardiac. Based on the appearance, two types of muscles are identified. Based on the appearance, two types of muscles: striated, non-striated, or smooth. Based on regulation, two types of muscles are identified: voluntary muscle and involuntary muscles. Muscles which are closely associated with the skeletal components in the body. Skeletal components of the body. Muscles which are closely associated with the skeletal components of the body. Skeletal muscles. Involuntary muscles of human. Smooth muscles and cardiac muscles. Smooth muscles and cardiac muscles. Voluntary muscles of humans. Skeletal muscles. Muscles are located in the inner walls of hollow visceral organ. Muscles located here are called visceral or smooth muscle or non-striated muscle. Muscles which are primarily involved in locomotory actions and changes of body posture are striated muscle or skeletal muscle. Striated or skeletal or voluntary muscles. Muscles which have a striped appearance under the microscope hence called striated muscles. Muscles which do not exhibit any striation and are smooth in appearance, hence they are called smooth muscles. Smooth muscles are non-striated muscles. Smooth muscle are non-striated muscle. Striated muscles of human, skeletal and cardiac muscles. Skeletal and cardiac muscles. Non-striated muscles of human, smooth muscles. Visceral muscles are smooth muscles. Their activities are under the voluntary control of the nervous system. Hence, they are known as voluntary muscles. Muscles which assist, for example, in the transportation of the food through the digestive tract and gametes through the genital tract are visceral or smooth muscles. Their activities are not under the voluntary control of the nervous system and are therefore known as involuntary muscles. Muscles of the heart are called as cardiac muscles. Muscles which are arranged in the form of bundles are skeletal muscles. Muscles which are arranged in the form of sheets are smooth muscles. Many dash muscle cells assemble in a branching pattern to form a cardiac, many cardiac muscle cells assemble in a branching pattern to form a cardiac muscles. Cardiac. Muscles whose muscle cells with multinucleated condition. Multinucleated condition is seen in skeletal muscle. Each organized skeletal muscle in our body is made of number of muscle bundles or fascicles, you can see. Bundles or fascicles. Okay. And held together by a common collagenous connective tissue layer called fascia. Each muscle bundle contain a number of muscle fibers. Each muscle fiber lined by the plasma membrane called sarcolemma and it encloses sarcoplasm. Muscle fiber is a syncytium. Muscle fiber is a syncytium as the sarcoplasm contains nuclei. Muscle fiber of skeletal muscle. Endoplasmic reticulum of muscle fiber is called sarcoplasmic reticulum. Cytoplasm of muscle fiber called sarcoplasm. Mitochondria of muscle fiber is called sarcosome. Storehouse of calcium ion in muscle fiber, sarcoplasmic reticulum. Nothing but endoplasmic reticulum. Sarcoplasmic reticulum is considered as storehouse of calcium ion. The storehouse of calcium ion in muscle fiber is sarcoplasmic reticulum. A characteristic feature of the muscle fiber is the presence of large number of parallelly arranged filaments in the sarcoplasm are called myofilaments or myofibrils. A detailed study of the myofibril has established that the striated appearance due to the distribution pattern of two important proteins. They are actin and myosin. Each myofibril has alternate dark and light bands. The dash bands, light bands contain actin. Light bands contain actin and is called I band or isotropic band, whereas the dark band is called A or anisotropic band contain myosin. Both the proteins like actin and myosin are arranged as rod-like structures parallel to each other and also to the longitudinal axis of the myofibril. Dash filaments are thinner. Actin filaments are thinner as compared to the myosin filaments, hence are commonly called thin and thick filaments respectively. In the center of each I band is an elastic fiber dash which bisects it. That elastic fiber in the center of I band is called Z line. The thin filaments are firmly attached to Z line. The thick filaments in the A band are also held together in the middle of this band by a thin fibrous membrane called M line. The A and I bands are arranged. A and I bands are arranged alternatively throughout the length of myofibril. The portion of the myofibril between the two successive Z lines is considered as functional unit of contraction and is called sarcomere. In a resting state, the edges of thin filaments on either sides of the thick filament partially overlap the free ends of the thick filament, leaving the central part of the thick filament. The central part of the thick filament not overlapped by thin filaments is called H zone. Chief fibrous protein of human body is collagen. That group of muscle bundles is called muscle. Group of muscle bundle is called muscle. Group of fascicles is called muscle. Group of fascicle is called muscle. Each fascicle contain muscle fibers. Bundle of muscle fibers can form fascicle. Bundle of muscle fiber can form fascicle. Plasma membrane will surround the group of myofibrils called sarcolemma. Multinucleate condition of the skeletal muscle fiber is also called syncytial condition. Syncytial condition. Large number of parallel filaments located in the sarcoplasm of skeletal muscle fiber are myofibrils. Structure with alternate dark and light bands in skeletal muscle fiber. Myofibril. Reason for the striated appearance of skeletal muscle fiber. Alternatively arranged dark and light bands of myofibril. Alternatively arranged dark and light bands of myofibrils. Reason for the striated appearance of myofibril. Distribution pattern of actin and myosin. Light band of myofibril contain actin, troponin, tropomyosin. Actin, troponin, tropomyosin. Dark band of myofibril is made up of myosin. I band contain only thin filaments. I band contain only thin filaments. Okay. A band except H zone contain. A band except H zone contain both thick and thin filaments. A band except H zone contain both thick and thin filaments. H zone contain only thick filaments. H zone contain only thick filaments. Functional unit of muscle contraction. Sarcomere. I band can be cut into two antimere passing this line. I band can be cut into two antimere passing this line. Z line. A band can be cut into two antimere passing through this line. M line. The central part of the A band without thin filament is called H zone. Each sarcomere contain half I band and full A band half I band, full A band, half I band. In sarcomere, thickermost part, A band except H zone. Thickermost part, A band except H zone, which contain both thick and thin filament. Thicker part, H zone, which contain only thick filament. Thin part, then within part is I band, which contain only thin filaments. In resting state, a part of sarcomere, a part of sarcomere with only thin filament, part of sarcomere with only thin filaments, I band. Part of sarcomere with only thick filament, H zone. Part of sarcomere with both thick and thin filament, A band except H zone. A band except H zone. Then in maximally contracted muscle, part of sarcomere with only thin filament. Part of sarcomere with only thin filament, I band. Part of sarcomere with only thick filament, H zone. Part of sarcomere with both thick and thin filament, A band except H zone. Wait. In a maximally contracted muscle, part of sarcomere only thin filament, I band, okay. Part of sarcomere with only thick filament, H zone, disappears. Actually, no zone at all. No H zone, we have to write. No H zone. That means our answer will be none. Then part of sarcomere with both thick and thin filament, A band, that's all. No H zone at all. In maximally contracted muscle, lengthwise, condition of I band, lengthwise condition of I band remains same. Condition of A band remains same. Condition of H zone disappears. Find out the changes in the given areas with respect to maximally contracted muscle in comparison to resting state. Length of actin molecule remains same. Length of myosin molecule remains same. Length of myofibril decreases. Length of sarcomere decreases. Find out the changes in the given areas with respect to maximally contracted muscle in comparison to resting state. A. Position of thin filament. A. Position of thin filament moves into H zone. B. Condition of Z line pulled towards A band. Each actin, that means thin filament, is made of two F or filamentous actins, helically wound each other. G actin means globular actin. F actin is a polymer of monomeric globular actin. Two filaments of another protein dash also run close to the F actin, that is tropomyosin. A complex protein dash is distributed at regular intervals on the tropomyosin, that is troponin. In the resting state, a subunit of troponin, a subunit of troponin, marks the active binding sites for myosin on the actin filament. Each myosin, that means thick filament, is also a polymerized protein. Many monomeric proteins called meromyosin. Many monomeric proteins called meromyosin constitute one thick filament. Each meromyosin has two important parts. One important part, a globular head, with a short arm and a tail. Heavy meromyosin include head and short arm. Light meromyosin include tail. The HMM, heavy meromyosin component, that is head and short arm, projects onwards. Head and short arm projects outwards at regular distance and angle from each other from the surface of a polymerized myosin filament is known as cross arm. The globular head of dash myosin is an active ATPase enzyme and has binding sites for ATP and actin. Mechanism of muscle contraction is best explained by sliding filament theory. Which states the contraction of a muscle fiber takes place by the sliding of the thin filaments over the thick filaments. Muscle contraction initiated by a signal sent by central nervous system via motor neuron. A dash neuron along with the muscle fiber connected to its constitute a motor unit. The junction between motor neuron and the sarcolemma of the muscle fiber is called neuromuscular junction. Or motor end plate. A neural signal reaching this junction releases a transmitter. Neural junction from this release a transmitter called acetylcholine, which generates an action potential in the sarcolemma. Action potential from sarcolemma spreads through the muscle fiber and causes the release of calcium ions into the sarcoplasm. Increase in calcium level leads to the binding of calcium with the subunit of troponin on actin filaments and thereby remove the masking of active sites for myosin. Utilizing the energy from ATP hydrolysis, the myosin head now binds to the exposed active sites on actin to form a cross bridge. Dash pulls the attached myosin head, pulls the attached actin filament towards the center of AMAN. Z line attached to these actin are also pulled inward, thereby causing a shortening of sarcomere. It is called contraction. It is clear from the above steps that during shortening of the muscle, the dash contraction of the bands get reduced, and whereas the bands retain the length. I band is getting reduced and A band retain its length. After power stroke, the myosin after power stroke, the myosin releasing ADP and phosphate ions, ADP and phosphate ions goes back to its relaxed state. A new ATP binds and the cross bridge is broken. The ATP is again hydrolyzed by the myosin head, and the cycle of cross bridge formation and breakage is repeated, causing further sliding. The process continues till the calcium ions. The process continues till the calcium ions are pumped back into the sarcoplasmic reticulum. Are pumped back to the sarcoplasmic reticulum, resulting in the masking of actin filament. This causes the return of jet line back to their original position. We call it as relaxation. Calcium ion has to go into the cisternae of sarcoplasmic reticulum. Reaction time of the fibers can vary in different muscles. Repeated activation of the muscle can lead to the accumulation of lactic acid due to anaerobic breakdown of glycogen in them, causing fatigue. Muscle contain a red color oxygen storing pigment called myoglobin. By mistake, we got answer here. Called myoglobin. Dash content is high in some of the muscles, which gives a reddish appearance. Such muscles are called myoglobin, and such muscles are called red muscle fibers. Dash muscles also contain plenty of mitochondria, which can utilize the large amount of oxygen stored in them for that ATP production. Red muscles also contain plenty of mitochondria, which can utilize the large amount of oxygen stored in them for ATP production. Some of the muscles possess very less quantity of myoglobin and therefore appear pale or whitish. Dash muscle therefore can also be called aerobic muscle, red muscle. Number of mitochondria are few in white muscle fiber. Dash muscles are with low amount of sarcoplasmic reticulum. Low amount of sarcoplasmic reticulum in red muscle. Dash muscle depends on anaerobic process of energy. White muscle. Dash muscle with high amount of sarcoplasmic reticulum. White muscle. Number of polypeptide units in each troponin are. Number of polypeptide units in each troponin are 3. Polypeptide units of troponin are TNT, TNI, TNC. Polypeptide unit of troponin which inhibits the myosin binding site on the actin, that is troponin I. The polypeptide unit of troponin which can bind with calcium ion, troponin C. Polypeptide unit of troponin which bind with tropomyosin, troponin T, TNT. Reason to consider the troponin and tropomyosin as regulatory protein because of their role in masking and unmasking of the active sites. unmasking of active sites. Reason for to consider the myosin as a motor protein because it is able to convert chemical energy into convert the chemical energy into convert the chemical energy in the ATP molecule into mechanical energy. Each myosin thick filament is a polymer. Number of polypeptide chains in each myosin molecule. 2. Arrangement of polypeptide chains in each myosin thick filament wrapped around each other. Wrapped around each other. Each monomeric protein of myosin is considered as meromyosin. Considered as meromyosin. Globular head with a short arm and a tail. The monomeric protein with the above parts. Meromyosin. Number of myosin molecules per thick filament. 200 to 300. Number of hemimeromycin for thick filament 400 to 600. Number of light meromycins for in thick filament 400 to 600. Motor protein of muscle which is able to convert the chemical energy in the ATP molecules into mechanical energy. Myosin. Each thick filament is a group of many monomeric proteins. True or false? True. Each myosin consists of two polypeptide chains and each polypeptide chain consists two monomers called heavy meromycin and light meromycin. True. One heavy meromycin, one light meromycin can form a myosin molecule. False. One heavy meromycin. This is false. One heavy meromycin, one light meromycin form a polypeptide and two such polypeptide can form a myosin molecule of 200 to 300 myosin molecules to form a thick filaments. True. Part of meromycin which serves as a flexible link between head and tail regions is neck or short arm. Myosin head is faced always towards the Z lines of sarcomere. True. Myosin head is faced always toward the head zone of sarcomere. False. Myosin can convert the chemical energy of the ATP molecules into mechanical energy. Hence, it is considered as motor proteins. Each myosin molecule is considered as a polymerized protein. True. Tail of myosin is always towards the Z line in sarcomere. False. Tail of myosin is always towards the H zone in sarcomere. Tail of myosin is always towards the H zone. True. Half of the myosin molecules have their heads oriented towards the Z membrane, other towards the H zone. False. Half of the myosin molecules have their heads oriented towards one Z membrane, other half towards the other Z membranes of the sarcomere. True. Tails of all myosin molecules in an A band are towards directed towards M line or H zone, we can say. Center of H zone is nothing but M line. The head and short arm project outward at regular distances and angles from each other to form the surface of a polymerized myosin filament. This phenomena is known as cross arm. Number of binding sites of each head of myosin are to binding site of myosin head are known as ATP binding site. ATP binding site and actin binding site. During muscle contraction, myosin head pulls the thin filament towards H zone, or we can say M line, center of H zone, M line. Invagination of sarcolemma into the sarcoplasm leads to the formation of T tubule, so-called transverse tubule, a motor neuron, and a set of muscle fibers innervated by all the telodendrites constitute motor unit. T tubule is non-mammalian vertebrate pass through Z line. Little non-NCERT here. T tubule in mammals penetrate and pass through these junctions. AI junction, just for information. It's not there in our NCERT text. The structures are flanked on either sides of the T tubule are terminal cisternae of sarcoplasmic reticulum. Terminal cisternae of sarcoplasmic reticulum. The junction between a motor neuron and the sarcolemma of a muscle fiber called neuromuscular junction or motor end plate. Functional unit of muscle contraction is sarcomere. The T tubule and terminal cisternae sides form triad system. Location of most of the stored calcium in sarcocyte. Most of the calcium in sarcocyte in terminal cisternae. Neuromuscular junction is also called motor end plate. Regulatory proteins of thin filament, troponin and tropomyosin. The thin fibrous membrane of A band. M line. Crucis membrane and H zone are also called Dobie's line, not there in NCERT. Dobie's line, which is also called Z line according to NCERT. Or we call it as Henson's disk. The theory which explains mechanism of muscle contraction in West way, sliding filament theory. Signals required for the initiation of skeletal muscle contraction generated at brain. Neurotransmitter which is released from motor neuron at neuromuscular junction to generate action potential in the sarcomere is acetylcholine. Depolarized membrane of sarcocyte due to the neurotransmitter released from motor neuron is sarcolemma. Cytoorganelle release calcium ion during depolarized state. Sarcoplasmic reticulum. Endoplasmic reticulum, here called sarcoplasmic reticulum. Sliding filament theory explained by Gene Hansen and Hughes Huxley. Brain sends signals to the muscles with the help of motor neuron. Sarcolemma transfer the information of muscle contraction to the cisternae of sarcoplasmic reticulum through transverse tubule. Calcium ion binds the TNC of the troponin of the thin filament. It results troponin and tropomyosin complex, tropomyosin complex to move away from the active site of actin molecule. Enzyme used for the activation of ATP of myosin head and the inorganic element who brings that enzyme towards the myosin head. ATPase and calcium. Actomyosin complex means binding of myosin head to the active site of actin. Troponin tropomyosin complex TNT binding with tropomyosin is called tropomyosin troponin complex. Cross bridge means actomyosin complex. The cross bridge pulls the attached actin filaments towards center of A band into H zone. Center of A band, which is nothing but H zone. When the thin filaments pulled inwards into A band, Z line also move inwards. Why? Why? Because of actin filaments. Because of actin filaments attached to H zone, attached to Z line. So it is important to note that myofilaments do not actually shorten, justify. The thin filaments are pulled into the H zone. The thin filaments are pulled into the H zone, making I band narrow, Hence, no alternation in the length of myofilaments. Myosin is changed into an active state so that it can perform the power stroke chemical reaction required for this action. ATP need to hydrolyze. ATP need to hydrolyze into ADP and phosphate ions. When myosin head binds to actin, it releases phosphate ion and undergoes a conformational change. Action performed by myosin head due to this change, pulling the thin filament towards the A band, nothing but H zone, we can say. When the power stroke is completed, myosin head release this molecule. Power stroke completed, myosin head release ADP, adenosine diphosphate. At the end of power stroke, myosin head binds to a new molecules of ATP. It results displacement of myosin from actin. Formation of actomyosin complex, power stroke, displacement of myosin head from one active site to another active site are collectively considered as cross bridge cycle. During relaxation of muscle, when motor impulses are stopped, changes happen with respect to sarcoplasmic reticulum and calcium ions are pumped back. Calcium ions are pumped back into the cisternae of sarcoplasmic reticulum. During relaxation of muscle, when motor impulses are stopped, calcium ions are back to cisternae. Changes happen with respect to thin filament. Masking of active sites of actin by troponin and tropomyosin complex. Troponin and tropomyosin complex. When motor impulses are stopped and calcium ion move back to cisternae, changes happen in muscle with respect to Z line. Z line go back. Z line go back to their relaxation. We can call here Z line go back to their original position. Repeated activation of the skeletal muscle can lead to fatigue chemical responsible for fatigue is lactic acid. Chemical released by skeletal muscle due to aerobic respiration, lactic acid. A. Utilization of stored ATP. B. Anaerobic respiration. C. Taking phosphate muscle phosphagen called phosphocreatine. D. Oxygen depth. E. Breakdown of glucose by oxygen. F. Pyruvic acid formation. G. Lactic acid formation. Arrange these in sequence with respect to the skeletal muscle contraction. A, C, E, D, B, F. G. This is order. Skeletal muscle fails to contract temporarily during fast muscle contraction due to the accumulation of this chemical substance, lactic acid. The lactic acid produced during rapid contractions of skeletal muscle under low availability of oxygen partly oxidized and major part of it is carried to and by liver through blood. Conversion of lactic acid into pyruvic acid and then the glucose to in liver is called gluconeogenesis. After the conversion lactic acid into glucose, liver send this glucose to and by glucose to muscle and by blood. In case if glucose is not immediately required, it can be used to rebuild reserve glycogen through this process, glycogenesis. The two-way traffic happen between the liver and muscle is called Cori cycle. Oxygen storing red color pigment of muscle is called myoglobin. Hemoglobin of muscle is called myoglobin. Cytoorganelle which is abundant in red muscle fiber, mitochondria. Muscle which are considered as aerobic muscles. Aerobic muscles are red muscles. Muscles with very low quantity of myoglobin are called white muscles. So read the statement and answer the form of true or false here. A. The heads and the two ends of the thick filaments are oriented in opposite direction to pull in actin filaments both the sides towards the M line. True. Head of myosin in myofibril is always towards the Z line. True. Tails of myosin in myofibril are always towards the H zone. True. Tails of myosin and myofibril are always towards the Dobbe's line. False. Write the statement and answer them in the form of true or false. Muscle contraction means pulling thin filaments towards M line. True. Myosin head contain Myosin head contain binding site of ATP and actin. True. In the maximally contracted muscle, length of I band decreases, length of A band remain constant. True. In the maximally contracted muscle, H zone disappears. True. Then read the statement and answer in the form of true or false. In the maximally contracted muscle, length of thin filament decrease and length of thick filament remain constant. False. Either contraction or relaxation, there is no change in the length of thick filaments. True. Crucis membrane is called jet line because of its jet-like appearance on electron microscope. True. Thickness wise, H zone is more thicker than the remaining A band. False. In myofibril, A band except H zone is more thicker than H zone and H zone is more thicker than I band. Yes, true. Sliding filament theory states that contraction of muscle fiber takes place by the sliding of thin filament over in between the thick filament. True. Sliding filament theory states that contraction of a muscle fiber takes place by the sliding of the thick filaments over in between the thin filaments. False. Troponin tropomyosin complex can be formed only during muscle contraction. False. Utilizing energy released from hydrolysis of ATP, myosin head now binds to the exposed active sites and the actin molecule to form a cross bridge and phosphate ion is released. True. Actin myosin complex can be formed only during muscle contraction. True. TNC calcium ion complex can be formed only during muscle contraction. True. Cross bridge pulls the thin filaments towards the H zone of A band is called contraction. True. During the shortening of the muscle, I band gets reduced in size or length. True. During the shortening of the muscle, the thin filament gets reduced in size or length. Muscles of the thin filaments get reduced. No. During muscle contraction, jet membrane of the sarcomere are brought closer. True. The combined power of several cross-plate cycles causes the muscles to contract. True. 225. Cross bridge cycle continues as long as the muscle receives stimuli. True. During recovery stroke, myosin head goes back to its relaxed state and release ADP. True. When a new ATP molecule when a new ATP molecule binds to the head of myosin, results the formation of new cross bridge. False. Read the statement and answer in the form of true or false. When ATP is hydrolyzed by the ATPase of the myosin, it leads to the cross bridge formation. True. During shortening of the muscle, the I band gets reduced in size. True. During the shortening of the muscle, A band retains in their size. True. So then we continue the skeletal system in the part 2. Till the time, thank you.